Ivy is a molecular biologist and a bioinformaticist who is now adding a third tool to her toolbox, which is differential equations modeling. And she'll be talking today about oscillations in gene expression, which is, Alex, I think you know something about that.
Great.
Thank you, Alan. I'm happy to be here. Can you see my slides all right? Now it's in full screen.
Yep.
Yes.
Yep.
Okay. Hello, everyone. I'm Ivy. So today I'm going to talk about my latest work titled Oncogenic Alterations in the PP3 pathway, Abolish Oscillatory Competence. So the story started 6 years ago when I was an exchange student at UCSD. Back then I was trained as a molecular cell biologist, as Alan introduced, and I was studying the role of a protein called cell4 in liver cancer. And here are some of the best images I took the cell I cultured. One day I was talking to Dr. John Watson at bioengineering, and I remember he asked, Ivy, what are the fundamental principles in cancer biology? I was embarrassed that I couldn't answer his question. And I felt there is actually a lack of principles. For example, there are great varieties of genetic changes that are associated with cancer. What's more, they are usually specific to one cancer type, which differ from one cancer type to another. Therefore, my research aims to address this problem. So I want to unify those genetic changes associated with cancer mechanistically. And today we're going to see how we can actually reconcile diverse oncogenic alterations in one of the key cancer hallmark pathway, the PP3 tumor suppressor pathway. So this pathway is a stress response pathway. So in response to stress such as DNA damage, the transcription factor PP3 would be activated, which in turn induce target gene expressions to facilitate cell decision, such as cell cycle arrest, apoptosis, or programmed cell death, and DNA repair. So these responses are important to control, to act against uncontrolled cell proliferation that suppress tumor formation. This pathway is the most perturbed in human cancers. For example, PP3 itself is mutated in over 50% of all tumors. And you can see here, almost all parts of the gene bodies are affected by somatic mutations. And there are 4 residues in the DNA binding domain that are most frequently affected. They're so-called hotspot mutations in PP3. In addition, many of the key regulators of PP3 are also commonly affected in cancer. For example, MDM2, MDM4, and ATM. And there are diverse alterations in this pathway. For example, inherited risk polymorphisms, somatic mutations, either truncating or missense mutations, loss of heterozygosity, copy number variation. It can be amplification, deletion of a gene, as well as epigenetic silencing. Therefore, my question here is, what is the common theme of these oncogenic alterations in the PP3 pathway? To answer this question, let's first have a look at how PP3 is regulated. So PP3 has a negative, has inhibitor MDM2, which is induced by PP3. MDM2 is a E3 ligase which attacks PP3 for ubiquitination and proteasome degradation. This inhibition and or negative feedback is so strong that without stress, PP3 is kept at very low levels in the cell. However, in response to DNA damage, this negative feedback relationship produces PP3 oscillations, as can be seen here in single cells, which can last for several days, with a characteristic period about 5.5 hours in human cells. So now you may ask, what is the functional role of PP3 oscillations? It turns out that PP3 oscillations are required for cell cycle arrest, and a switch from oscillations to a high dose of PP3 marks the escape from cell cycle arrest. In contrast, sustained high dose of PP3 commits cell rapidly into senescence or dormancy. Therefore, we have seen that PP3 oscillations are essential for cell cycle arrest and DNA repair. Therefore, I reasoned impaired PP3 oscillations would compromise genome integrity and promote cancer. Is it possible that the oncogenic alterations in the pathway would actually impair PP3 oscillations? To test this idea, I constructed a ODE model of the PP3 MDM2 system. So this ODE model describes the core negative feedback relationship between PP3 and MDM2. So the model described the production rate, PP3 is produced at a constant rate alpha, and is degraded by binding to MDM2 with a degradation rate beta. MDM2 transcript is induced by PP3 with a transcription rate sigma and degraded with a rate of eta. MDM2 protein is produced proportional to the transcript level and is degraded with a degradation rate delta. I chose appropriate parameter values based on previous calibration, and the model simulation produced oscillatory response in the system that is consistent with the experimental observations. And you can see here the state space trajectory of the system represent a limit cycle trajectory with the preferred frequency and is robust to noise and invariant to initial conditions. Then I reviewed a wide range of oncogenic alterations in this pathway, and I found that they affect full parameters in the model. For example, truncating mutations, allelic deletions, or inherited cancer risk polymorphism in PP3 reduce the production rate of PP3 alpha. Hotspot mutation in PP3 reduce the degradation rate of PP3 beta, but loss of ATM overexpression of MDM2 both increase beta. Also, PP3 missense mutation and overexpression of MDM4 would decrease the transcription rate of MDM2 sigma here. But inherited polymorphism in MDM2 or genetic amplification of MDM2 would increase sigma. Also, loss of ATM would decrease the degradation rate of MDM2 delta. I want to point out that while some of the changes affect a single parameter, there are oncogenic changes that affect multiple parameters simultaneously, such as loss of ATM here. First, let's have a look how sensitive the system is to value of a single parameter. As shown here, a 50% reduction of PP3 production rate alpha would produce dampened oscillation. This means that losing a copy of PP3 would lead to haploinsufficiency. And it's clear from the heat map here, lowering alpha reduced initial PP3 pulses, and it would diminish the oscillatory behavior of the system over time. Lowering delta, the degradation rate of MDM2 has a similar effect. lowering beta, the degradation rate of PP3, or sigma, the transcription rate of MDM2, markedly enhance initial pulses and ultimately cause a loss of PP3 oscillations. On the other hand, if you increase beta or sigma, the initial pulses would be reduced and the oscillatory behavior will be diminished as well. In terms of long-term behavior, PP3 oscillation is sustained only when alpha is sufficiently large. For delta, beta, and sigma, we can see here that PP3 oscillation emerge and disappear as the parameter value changed. This suggests a Goldilocks effect, meaning that PP3 oscillations are viable only within a narrow range of parameter values. Now that we have seen that each of those 4 parameters can affect the oscillatory behavior of the system, how about multiple parameters? And are there qualitative changes? In other words, was it a bifurcation from a stable equilibrium point to a limit cycle attractor at the back? So this question can be addressed by applying the Hopf bifurcation theorem, which tells us if a pair of complex conjugate eigenvalues passes the imaginary axis as the parameter passes a critical value, then the equilibrium point becomes unstable and the limit cycle attractor is created. For example, as the parameter C in the demo here reduced from 1.1 to 0.9, the eigenvalue evaluated at the equilibrium point changed from having a negative real part to a positive real part. This is accompanied by a bifurcation from a stable equilibrium point to sustained oscillations. Therefore, I performed a Hopf bifurcation analysis in my model. First, I tried to solve the equilibrium point. So at the equilibrium point, we have the derivative of all 3 variables equal to 0. Therefore, I can derive the equilibrium solution analytically in terms of the model parameters. Next, I performed the linear stability analysis, and the Jacobian matrix evaluated at the equilibrium point is shown here. The eigenvalue of the Jacobian matrix are solution to this characteristic cubic equation. Taking the expression from the cubic equation, now we can express the eigenvalue in terms of P and Q, which are themselves functions of model parameters. Notice that if these eigenvalues are complex conjugate, the real part will be minus P over 2. Therefore, now we have the Hopf criteria for sustained PP3 oscillation. First, P changes from being positive to negative so that the real part becomes positive. And second, P squared minus 4Q is negative, so the imaginary part exists. Using this criteria, now we can investigate how changes in multiple parameters would affect system behavior. First, let's have a look at how ATM triggers PP3 oscillations. In response to DNA double-strand breaks, ATM is activated. ATM in turn activates PP3 oscillations through 2 actions. First, ATM blocks the MDM2-mediated degradation of PP3, thus decrease beta. Second, activated ATM also accelerates the degradation of MDM2, thus increasing delta. I evaluated the Hopf criteria by varying these 2 parameters, and I found that sustained PP3 oscillations are viable in a narrow island in the beta-delta parameter space. The red dot here marks the best fit parameter combinations during DNA damage response. Therefore, the actions of ATM in triggering PV3 oscillations can be explained as a Hopf bifurcation, shifting the system from the green region into the Hopf bifurcation, the Hopf region here. And in this context, a loss of ATM would keep the system out of the Hopf region, as indicated by the dashed arrow here. Next, we can evaluate all the oncogenic alterations we have reviewed earlier by evaluating the Hopf criteria by varying all the 4 parameters affected by the oncogenic alterations. That is the production rate of PV3 alpha, the degradation rate of PV3 beta, the transcription rates of MDMT transcript sigma, as well as the degradation rate of MDMT delta. Considering beta and delta always appear as a product, we can combine them together. So now we can see Hopf bifurcation in a 3D parameter space. So when I evaluated the Hopf criteria, I realized that PV3 oscillation are sustained only in a subspace that is enclosed by the gray surface here. And the red sphere denote the best fit parameter combinations during DNA damage response. Notice how close it's situated relative to the boundary, which means that a slight shift in parameter values would trigger a qualitative change in system behavior. So in this dynamical framework, there are 4 routes for diverse oncogenic alterations to abolish PV3 oscillations. First, PV3 missense mutations reduce sigma. Some hotspot mutation also reduce beta. These mutations would push the system out of the Hopf subspace along the beta sigma axis to the left. Second, loss of PV3 reduce the production rate of PV3 alpha, thus dropping the system out of the Hopf subspace. Third, overexpression of MDM2 increase the transcription rate sigma. Overexpression of MDM2 has a net effect of increasing the product of beta and sigma. Therefore, both changes would draw the system out of the Hopf subspace along the beta sigma axis to the right. Lastly, we have already seen that a loss of ATM would keep the system out of the Hopf subspace on the beta sigma delta plane. Now it is clear that despite all those diverse changes, they have a common effect. By drawing the system out of the Hopf subspace, they all act to abolish the oscillatory competence of PV3. And here is a 3D view of the Hopf subspace. Notice that different changes are pulling the system out of the Hopf subspace along different axes and different directions. And notice how close the red sphere is relative to the surface. In summary, in this talk, I have shown that in response to DNA damage under normal physiology, PV3 oscillations are critical to cell cycle arrest and DNA repair. However, oncogenic alterations abolish PV3 oscillations, leading to accumulation of DNA damage, therefore leading to genome instability and aneuploidy, which ultimately promote cancer. With this, I'd like to thank you for your attention And I'd love to take questions.
Wow. May I ask a question? Couple?
Sure.
Ivy, that was a great presentation. Very clear, very well done. Wow. Let me ask you, is MDM4 the same as MDMX, that Jeff Wall discovered?
That's a different name for the same protein.
So you don't have MDM4 in your model?
We have implemented, just 1 second.
Yeah.
To exit. So we have implemented through different parameters, if I put it here. So MDM4 is one of the inhibitor of PB3. So its effect is acted through modulating beta and sigma simultaneously. So how I studied it was to evaluate, okay, how the level of MDM4 would change the parameter in terms of beta and sigma here. So it's like the effect is funneled through the change of parameter values.
Okay. So I worked with Jeff Wall about a decade and a half ago, and we actually produced a model which includes MDM4 dimerized with MDM2 in that process. And we also included constitutive degradation of the molecules in your model. Now, that's probably small, but you don't have it in any of these. You made the assumption that constituent degradation, for example, of, can you show your equations again?
Oh, yeah, sure. Oh, here.
Yeah, okay. So like in the P53 equation, alpha is constituent production, beta YX is your MDM2 action on P53.
Yeah.
Right, and you don't have any like linear constant times P53 degradation. You're assuming that's to be?
Yeah, so we have simplified the model, which we assume that degradation mediated by MDM2 are the major factors because of the strong negative feedback.
Yeah, no, and in fact, I've always suspected that that's the case because MDM2 really is the dominant factor that's making this go. However, when you begin to add variables and begin to get oscillations, then the system gets a little bit more complicated, and these small factors then might have some significant effect. So eventually what we did do and did not publish yet because we haven't completed the model. I'm actually working with someone here now, Amina, and we've added WIP1 to this, which is the phosphatase that is involved with virtually all of these molecules. And we're trying to see if we could get our normal cell data, which is skin cell data. We actually have a lot of data, and we've fitted the models, and the cancer cell model fits the data very well. And it does have oscillations still. They're decaying oscillations, but then again, we only put a single pulse in, and the data is whole cell data, so they're probably out of synchrony. Well, when are you going to publish this? Because I think it's exciting.
Yeah, we submitted already.
Oh, you did? Okay, that's great. That's great. Well, can I just bring your attention to where I do have some of this published, at least for the cancer cell data, and you might take a look at it and tell me what's right or wrong about it from your point of view.
I'd love to.
Yeah, it's in my textbook in chapter 2 chapters.
I'll note it down.
two chapters, 15 and 17. There's a lot of details there, including some of the data. We didn't publish it in the paper yet because we haven't really got it fixed for normal cell data, and I don't think it's right to do until we manage that. But I can even send you copies of this chapter if you like.
Oh, thank you very much. Yeah, I'd love to
Okay.
read it.
And I'd
And
like
check
to,
with my model.
I'd like to get your response, because you have a fantastic handle on this. I'm really impressed.
Yeah, I also really enjoyed your talk, Ivy. It was really, and the work is fantastic, but the delivery also was super clear. My question is regarding the initial input. So DNA damage is what triggers the dynamic response, and the degree to which DNA damage occurs, and also potentially the DNA damage signal itself may have a dynamic, maybe dynamically modulated, right? Where there's, and I wonder whether you explored that.
Yeah, so that's one of the hot topic in the field right now. So how would the dynamic differ when you have different kind of external driving force, right?
Exactly.
So here we are pretty much interested to see like how the PB3 oscillation started and how a different oncogenic changes would actually affect the capability for them to for the molecule to oscillate. But the next question would be how different external input would alter the dynamics. And one of the best examples actually coming out from in a kappa B system is if you drive it with a different, like a strong enough input, the oscillation might change into like a chaotic behavior. And this, I think, like it's also interesting to see whether we can replicate in PV3 and what kind of outcome that will be. I think that's my next step of research.
That's great. Yeah. So we study NF-κB in my lab, which is why I'm asking the question.
In fact,
then you must have read this paper. It's beautiful.
Yeah. So can I ask one more question about that? After actually a follow-up of Alex's. So yeah. I recall about a decade ago some work from Harvard where single cell breaks. This generates double strand.
Right.
Single strand breaks generate no oscillations, right? They just get one big hump, and
yeah. So that's the case when we were looking at the data here. So on the right hand side, you can see like there are sustained PV3, like high level of PV3, and for the case of ultraviolet radiation, which is mediated through ATR, we only see kind of sustained like high dose of PV3. Mm-hmm. Actually, our model can explain it mechanistically, because the difference in terms of ATM mediated response and ATR mediated response is how they change the parameter values. So ATR only mediates or modulates the value of beta here, which is degradation rate of PV3, but it does not change the degradation rate of MDM2. So what it does is it would push the system to the left. Instead of pushing it to the northwest, it pushes it to the west, which would be way harder to reach the Hopf region. But at the same time, the equilibrium point, the equilibrium solution tends to have a higher value. So that pretty much explained why we tend to see a sustained high dose of PV3, but not oscillation when it's induced by ultraviolet radiation instead of the double strand break, which in the case for the single strand break. I hope that answers your question.
Yes.
Great.